Yes. Okay. So welcome everybody to session 2 of this workshop on science and technology for the new age, acquisition, analysis, and adaptation. We had a very lively session in the morning where we had a plenary talk from Google, who talked about the various climatic calamities and the climatic changes that we need to make in our habits to ensure we have a brighter future for our children. We also had a very nice talk from Professor Jayashree Sabrinathan, who talked about multispectral imaging techniques, and was followed up finally by Professor Scott Chang, who talked about soil and greenhouse gas mitigation efforts that his research work is currently doing. So this session is mainly about, plants and how they sort of energy and the climate sciences. And we have one beautiful tutorial lined up to be taken by Professor Yashwanti Mudgil of the Department of Botany, Delhi University, India. She'll talk on introduction to phenotyping through stress interactomes in Arabidopsis thaliana. She's been a collaborator with us for a long time, and we are looking forward to her talk. The second talk will be by Professor Sudeshna Chattopadhyay from the Department of Physics, IIT Indore. She will talk about rechargeable thin film batteries and the effects of, and talk about the physics of interface engineering that are useful for building such devices into portable batteries and finally putting them into sensors and things like that. And then we have a talk by Logi Ag, who will talk about the applications of libs. Unfortunately, they're yet to send the title, but they have confirmed that they'll be here. And the final talk of today's session will be by a PhD candidate, Balsher Sidhu of the University of British Columbia, Canada, who will talk about a very interesting topic of the impact on climate variability on crop yields in India. Since the first talk, as I mentioned, will be by Professor Yash Mudgil. And just to introduce the speaker, so she has been working as a faculty member at the Botany Department, University of Delhi. And she obtained a doctoral degree from the Jawaharlal Nehru University, a very famous central university in India. And then she did a postdoctoral work in the University of Toronto and the University of North Carolina at Chapel Hill, USA. She has also been a visiting faculty at the University of North Carolina on a Department of Biotechnology award. And her current research interests include molecular characterization of stress-mediated signaling pathways in plant cells. So with that, I request Professor Yashwanti Mudgil to start her presentation. And may I request all the participants to please mute themselves and refrain from asking questions. She has a 50-minute talk, and then we have 10 minutes for question and answers. So you can feel free to type your questions in the chat box, and we'll take all the questions at the end. Thank you for your cooperation, and over to Professor Mudgil. Thank you.
Thank you, Amrita. I'm delighted to be part of this nice workshop, and which I think the topic is very nice. Like, it's pretty broad, which encompasses kind of all walks of kind of students, postdoc, industry, and everybody kind of comes under one umbrella. So thanks for giving me opportunity to participate in this and to be part of it. Okay, so let me get started with my presentation. Is it visible to all of you?
Yes,
we can see it.
So welcome to my presentation, and the title of my talk is Molecular Characterization of Stress Interactome of NTL1 AGV module in Arabidopsis thaliana. So as this is a tutorial, I'm going to go over the major kind of work we are doing to solve an issue which is a big issue to humankind. Okay, so we all know that climate change and the agriculture is greatly affected by it, right? And it's a huge problem to farmers, and it is reported in 2017 in PNAS report that more than 59,000 farmers committed suicide in India since 1980 due to this adverse climate change effects. And the temperature is rising constantly, right? And this is a big problem in the sense that with every degree increase in temperature, we are going to get lesser and lesser yield. And here the example of rice being given. So the total overall 8.3% less yield would be there if there is an increase in temperature by 3 to 6 degrees. And this just to to kind of give you an overview that how food insecurity and climate change are coupled to each other, right? So if we don't have like we can't provide food security because we are losing the crops, right? So the overall yield is decreasing. So here you can see with this scale that majorly you can see even India is also affected, right? We are in moderate range in between. And obviously you can see the Africa and the other parts also, they are severely affected by the food insecurity coupled with the climate change. And if we try to see this in terms of saline salt stress, the common arid and semi-arid regions where evaporation exceeds precipitation, it results in salt accumulation in the soil, and definitely it would lead to reduction in the productivity. And you could see here with the help of this heat map that various parts of our world are affected with various degree, and definitely like you could see the mostly the it shows the desert regions, but also the other part also affected by accumulation of this saline soil, and which definitely in turn would lead to the decrease in the crop yield. If I talk specifically about specific abiotic stresses, if we talk about heat, it results in severe problems in case of major stable food crops. Like here, I'm giving example of wheat, rice, and corn, and the sugarcane as well. So it results in reduced germination and total overall protein content, and reduced yield overall in all of the crops. And in terms of water shortage, again, these three, including the cotton, they are severely affected, and reduce rate of leaf appearance and area and yield is kind of again affecting all these crops. Salinity also affects, and here cotton is replaced by tomato, and you could see that it lead to severe reduction in the yield, as well as you have reduced germination due to salinity, and shoot length and fresh weight of overall vegetative mass of all these plants decrease. So all these abiotic stresses, they in terms of physiological processes, if you try to see, they are affecting stomatal conductance, photosynthetic rate, photosystem 2, membrane fluidity, and pigment and gene expression, and resulting in they are affecting the flower, fruit, seed germination, leaf area, root architecture, and plant height, and ultimately the yield is affected, right? So what are plant going to do? They definitely, being live, they are going to fight back, right? It's a normal feature of all of us. If we are stressed, we need to fight back. So they are also fighting back, but different plants have different stress tolerance potential depending upon their genetic constitution. So their fighting abilities are different, right? So there are some which can survive in the salt, right? And in high concentration of salt, some can't, right? So it's all in their genetic constitution. So when plants are experiencing all these stresses, they tend to overcome it by changing their physiological or metabolic processes which are going within, and which in turn it definitely affects their growth and development. For example, let's say now plant was happy, no stress was there, but now it is stressed, so it will not make seeds. It will try to save its life, and so it's changing its physiology, it's changing its growth pattern, and overall growth is affected in that sense. So there are various reports, and agriculture sector is going to be highly impacted due to all these changing climate situation, and we need to think about strategy that how are we going to cope with these situations. So there are like what is my interest, how I am pursuing this fight. So in plants, or as well as an animal, we have I'm talking about this process at the cellular level now. Okay, so when we talk about a cell, on the cell surface we have receptors, and you on receptor you have heterotrimeric G proteins are coupled to those receptors, and this is what I'm showing is an animal model. Okay, so you see these black 7 transmembrane receptor structure. Then you have heterotrimeric G protein, alpha, beta, and gamma subunit. So when you have any stress or any other effector comes and bind to these receptor molecule, it would lead to activation of this receptor. And this would be nothing but change in the conformation of the proteins. These are nothing but proteins, right? So something is binding to the protein, it will lead to the structural conformational changes, and the alpha beta gamma subunit will beta gamma subunit will dissociate from the alpha subunit, and now they move inside the cell, and they will be interacting with other partners or the effectors, and they will go inside the nucleus and tell the cell, okay, look, some stress is there, you need to do something, right? So those downstream effector, they are there are layers. So you have first degree on the surface, then you have secondary messenger, then you have one more layer, and ultimately the gene expression would be change in physiology or change in the phenotype, right? So this is the simple the upper model is from animal, and in plants also G protein components are very simple in structure compared to animal, because animal has all these components are in multiple numbers in case of animal, whereas in case of plant, I'm taking model organism Arabidopsis example. So you have there is no G protein coupled receptor reported so far, but there is a similar protein RGS1 which is reported, and then you have alpha subunit just one, beta gamma, beta only one, and three gamma subunits. Okay, and effectors have been isolated. Now more and more information is pouring in, but only very few has been characterized in molecular details. So as I was saying, G protein signaling is very important for animals also. So or any biological process, like any physiological process you think of, they are involved in hypertension, diabetes, and uh, like your touch, your taste, and all these neurotransmission, they play very important role. And because of this, they are a huge market in case of pharmaceuticals, because people are looking for the drugs to target these and design the drugs. Okay, so it is from that perspective also G protein signaling is very important. So the organism which I'm working with is Arabidopsis thaliana model plant, and it definitely it has been well studied and established that it regulates seed germination. The G protein is doing important function in all these biological processes, regulates seed germination, cell expansion, division, ion challenge regulation, and we have chosen this because this is a very fast growing and plant. You can have the complete life cycle in 40 to 60 days from seed set to the next generation seed set. And whole genome is sequenced. Genetic manipulations are very, very easy, and knockout in almost all the genes of these plants are available. And definitely once you set up something that you find some genes with improved agronomical traits, you can use this information to the crop plants. So what are we basically doing? What is our strategy? How are we planning things in lab to fight back the abiotic stresses in plants? So my story starts with the way I showed you alpha, beta, and gamma subunits. So my story starts with the beta subunit. So beta subunit is already reported since 2000, I think, 2003, it was reported for the first time in Arabidopsis, and it plays very important role during the plant growth. As you could see, when you knock this protein out, the seedlings are shorter, and but interestingly, in root they have huge mass. So you have lot lateral root formation, and the root mass is huge. Leaf also have the round and short compared to the control wild type plant, and stomatal density is also different. So these are the important traits to begin with, which kind of attracted me, and I got interested into it from the stress point of view also. This stress information is more of kind of recent. More and more information is being added that this is playing very important role in stress signaling also. So you could see that the upper panel you see calls here, which is the control plant without any gene being knocked out, and AGB1-2 is the plant where you have knocked out the beta subunit of G protein signaling. Now, when you provide drought stress, you could see that the wild type normal plant can't survive, but beta is also stressed, but you can see more greening here, right? They are still able to survive better. So it has improved drought tolerance. And on the right hand side, you can see in terms of salt stress also. Before the upper one is the without salt treatment, and the lower one is after salt treatment. So when you knock this gene out, they are not able to withstand salt stress. So it has opposite effect. So in case of drought, it is able to withstand, and whereas in case of salt, it is not able to withstand. So the same gene is doing different function and different stress, but it is playing very important role. And similarly, people have studied that germination and other phenomena also, which I'm not going into details, but all those phenotypes are also there in case of beta mutant. And if you try to see in terms of reactive oxygen species, they are lower down in this mutant, which can be measured in terms of peroxidase activity. And when plant is stressed, they increase certain amount of proteins or sugars to fight back, right? So their osmotic milieu is changed in response to that. So the proline content was also found to be lower in case of beta mutant compared to the wild type. So all these kind of things made me interested into the beta, and I pursued beta signaling. And to begin with, I started with the what I'm interested in going downward in the cell. What is happening? What is beta activating further? With what it is interacting, and how it is working at the molecular level. So we performed a yeast-2 hybrid or yeast-3 hybrid screen, where you simply can understand that you take a protein and you use it to screen a library where all the proteins are present. So if it binds to certain protein, if it interacts with certain protein, it will bind to those, and it will result in expression of a certain reporter, which you can score, and you can know, oh, this interaction was successful, and then you have found that interactor. So that's what is, in very simple word, is to hybrid screen. So you can, using this technique, it's a very old technique and very beautiful technique, but it's still very favorable technique to screen like the interactor in the beginning. There are a lot of false positive also, but for the first go, it is pretty robust. Like you can select some, and then you can further characterize those whether they are real or not. Okay, so I used 3 hybrid in the sense because I needed beta gamma dimer. I was not looking for the 2 hybrid. 2 hybrid is when 1:1 protein interaction is there, but here it is, I'm using 2 protein, beta and gamma dimer, and I'm looking for its interactor. So I did that screen, and I found a lot of interactors. That was my postdoc work. So I chose some, and this technique can be, let's say, if you are working in, you are interested in studying gene X or protein X. So you can use the same strategy. You can use the yeast-2 hybrid. You can make the library, means the representation of all the cDNA of that organism, and you can screen using the weight which you are interested in. So this technique can be used for any kind of plant or any kind of organism. It's kind of universal. Everybody uses it, people in animal as well as in plants. So I found certain interesting candidates, which I followed up further, and NDL1 was one of them. So in the beta screen, I found NDL1, and I pursued it further. So what is NDL1? So it is a beta interactor, of course, because we found it in the beta screen. And if you can hook up any gene with any reporter, it could be a biochemical, it could be a chromogenic reporter, that which when you add, it could be an enzyme. If you add substrate, it will give you color, or it could be a fluorescent protein, right? So you can make fusion proteins using either GUS or GFP. And then you can study the detailed localization of that particular protein you are interested in. So we use the same approach, and we analyzed in detail where does NDL1 localizes in the plant. And we found that it does share a lot of localization pattern with beta, with which it interacts. Not all, but some places. So it could be that it interacts with beta and do some function in growth and development of that organ, but not everywhere, right? So which is very normal. And so we found the expression in root, majorly root, and in the pollens as well. And we it's positively regulate primary tooth length. We did a lot of studies and established all these things. So positive regulator of meristem initiation, and it responds to various hormones also. And recently we found, and another group also have studied and found that they could be used as stress marker also, because they have shown expression pattern changes in presence of various kind of stresses. So the way beta mutant has been studied, the same way we did the knockout analysis in case of NDL1. So it does lead to the various vegetative defects if you knock these protein out. And you will have the fused flowers or kind of abnormal seed distribution in the plant. And you can further do in silico analysis coupled with the in vivo analysis and plant analysis. You can study there are various tools available now that because the way I said, because the whole genome is sequenced, a lot of people do expression experiment using microarray analysis, and they deposit those data into the databases in response to various stress, in response to various mutant. So the huge information is available for this model, and you can easily, whatever protein you're interested or gene you're interested in, you can see where it is expressed from the other people's experiment as well as from yours. So you can do all those analysis from existing information. So there are three members in this gene family, 1, 2, 3, and you could see that they develop during the different stages of development, their expression pattern varies. So 1 and 2, 1 and 3 share more closer pattern compared to the 2, which is little different in different tissues of the plant. And then that was the in silico approach. Then we did in vivo approach also. The way I told you that you can make fusion with anything, we made Gus fusion with the promoter, and we studied the expression in different parts of the plant during different stages of development, 4, 8, and 12 days. And even because we discovered it as interactor of beta, we did expression presence and absence of beta during different developmental stages. And but our main goal was to we are interested in abiotic stress signaling, right? Because we want to find some candidate genes which would be kind of key genes and can be used later on in agronomic trait improvement. So with that aim, we have chosen beta and NDL1, right? So we studied like in response of this protein to various kind of abiotic stresses and hormones also. And we did see that there is increased localization after different hormones, which are considered to be the stress hormone, like salicylic acid, jasmonic acid, and even with cold and PEG and heat, like various treatments were used, and all three members were analyzed. NDL1, this is for 1, and you could see that PEG treatment lead to the increase in Gus staining. So the protein amount increases when you do PEG treatment, right? And for NDL2, you could see that the various abiotic and hormonal treatments are given again, and you could see that it's other way around. NDL3, during the no stress condition, the Gus staining is there, but when you treat with mannitol and PEG, which are nothing but you are giving the dehydration stress, right? So when you put mannitol or PEG, the plant will ooze out the water, so it will create a dehydration stress like situation for it. So there was no change under cold treatment or ABA and GA, but definitely with mannitol and PEG, there was a reduction. Protein amount was less compared to the wild type situation. We tested for 3 also, and we did in presence and absence of beta also. So all these treatments ultimately showed us that this gene family is stress responsive, and majorly in case of dehydration stress, we found that this is for NDL3, and you could see that when no stress is given, almost no protein is present. But when you give the mannitol and the PEG treatment, then your staining is increasing, right? So what we concluded out of all these studies that uh, NDL1 localization is upregulated during cold, IA, J, and SA, and in response to the cytokinin treatment also, it was downregulating after heat stress also. And NDL2 expression was upregulated after ABA, cold, and J, but no difference in the mutant background. So it might be functioning independent of beta there, right? So, and 3 expression was unaffected by various kind of hormones and heat treatment also. And all these results support that NDL1 and 3 definitely have a role during the dehydration stress or the drought stress response. And 2 might be playing ABA dependent indirect role. It might not be direct effect. So we, after establishing all this, that okay, now we know beta was already established. Now we know that where NDL1 expresses, whether it shares expression with beta and responds to abiotic stress with beta. After doing all those studies, now we know that G protein as well as NDLs, they are playing important role in the growth and development as well as in doing abiotic and biotic stresses also. I'm not speaking about biotic stresses, but we have found some data where we can kind of find that biotic stresses are also combated by this duo. So especially beta subunit and 1 and 3, they have a kind of a special role in case of dehydration stress. So now this we are talking about the independent unit, but at the simultaneously G protein interactome has been solved. I was part of that initiative also. I had opportunity to participate in that screen. That what was done, that all the subunits of G protein were taken, and okay, so now nowadays, like the way I was saying that all these things are in layer. You can go from gene level, you can go from protein level, or you can go from RNA level, right? So proteomics is kind of more still evolving, but in terms of techniques, but it is more informative. So use people are using interactome approach, where you can use the large scale E2 hybrid or protein protein interaction approaches and find out like the whole interactome of an organism under a given situation. So that was done for G protein, that various subunits were taken, alpha, beta, gamma, and NDL1 was also taken, and few more. And the whole interactome of all those proteins were purified from Arabidopsis. And it was found that NDL1 interacts with 62 proteins. And when this interaction, we know that the major problem is that we want to find out interactor which are fighting in abiotic stresses. And okay, we started with something which itself is involved in abiotic stress management. So we were hoping that we will find a lot of interactors which are involved in abiotic stress, right? And so that we can find out the component which help the plant to survive better under and to cope with the various agronomic losses, which can help us to manipulate all those things. So in current situation, we have a lot of information that interactome of AG1 and DL1 duo, it is definitely has a lot of components which can help us to figure out a couple of things and even to look for some key component. So what I decided, because root is a well accepted model for grow, right? For the whole plant to grow, you need more time, and these are pretty easier and faster assays to do. You can just grow plants on plates, as Arabidopsis is maintained in the culture room. You don't need to, like very huge infrastructure, so it's easily grown in the petri dishes. So we started with the further approach that to search for the effector of this module and to determine the function of those effector, definitely in abiotic stresses, and to find out their connection to the specific stress-related processes. So the way I told you that I started with the beta interactors. I found NDL1, right? And now I'm down here. So what we are doing we are moving from surface to inward, right? So we have moved after signal perception, beta gamma will separate, it will go downstream and it will interact with other protein, that was NDL1. Now NDL1 will interact with other proteins and ultimately regulate gene expression. So we are at this step now, okay? So this whole interactome was solved, and 62, the way I said, 62 interactor of NDL1 were there, and out of all 62, more than 50%, they are involved in either in biotic or abiotic stresses. So we pursued those interactions or interacted further, and we like our detailed in silico analysis and the literature searches tell us that they all are involved in or their putative function are in cell differentiation, cell fate speciation, cell cycle, and protein folding, and all those kind of functions they perform. And we are taking like approach to find out that whether these effectors they are playing role in abiotic and biotic stress and root development, and to confirm their genetic and physical interaction with the G protein core members, and characterization of role of these effector in stress and later root formation as well, and detailed characterization of these effectors. So the way I said, there are total 62 effectors. So this is just showing you the interactome of NDL1, 62, all 62 of those are there. And because the other proteins were also used, so this graph just shows that we were able to find 24 interactor of stress-related interactor of GPA1, 23 for beta, and 7, 6, and 1 for gamma subunits. And there were 31 for the NDL1. And when you do in silico analysis, they show response in terms of expression analysis to various abiotic stresses. And this is just the putative root and abiotic stress related interactor we have selected for study in the lab. And the way I was saying that there are different layers of regulation. You have genome, then you have proteome, then you have metabolome, because these proteins will interact with each other and ultimately they will regulate the metabolism of the cell, right? And then you will have biochemical reactions occurring, and those are further down there, right? And so we are at the step of protein-protein interaction when you take those protein out. So E to average was there, right? So we did that part. Now we are moving downwards. So again, in layers, you can see the physiological phenotypes, germination, growth, or flowering at the upper level. Those are morphological, visible phenotype you could analyze. But then you will have the downward metabolic networks are there, which you have to do further analysis, right? And then you can have the protein network, and then you can have the gene expression network down there. So you can easily do the gene analysis from in silico analysis, that a lot of data is being deposited. You can pick up any gene ID number and you can search for it. You would know where it is expressed in plants. So huge RNA expression profiles are available. So, and even you can assign the process to which they must be involved. So you can easily take gene of your interest or protein of your interest, and you can find out the prediction in which network it might be, process it might be involved, or which kind of function it might do. So we have done that for all these 31 NDL1 stress-related interactor, and we can find that a lot of them are involved in enzymatic activity or protein binding or metal binding kind of function, and which kind of this study corroborates with the animal studies as well. So you can do, there are a lot of nice tools available. This is one of those, the EFP browser. You can look for the stress people have done, like when people are doing microarray, they are studying the expression profile of the whole set of gene present in the organism. So your gene must also be there. And when the whole data is deposited for expression, your data is there. So you can do the data mining from the microarray data in the public domain, and you can find out. And that's what we did. This is from the different developmental stages. For some of the selected candidate, you can know like where is the highest expression, and some of the candidate we are interested in, we have chosen. And then you can find the abiotic stress specific expression pattern also in response to, let's say we have taken 3 candidates here. You can see in how do they behave in terms of gene expression in response to heat, ABA, cold, drought, or oxygen. Okay, and you can know those values, and based on that you can plan your physiological experiments, right? So these are some of the selected candidates we are interested in. And so the way I was saying, first you you isolated those from yeast to hybrid. Now you want to know whether in real are there, because there are a lot of false positives there, not a lot of, but you have possibility of false positives there, right? So you will first try to make sure whatever you have isolated, that is the real interaction. So there are techniques where you can split the fluorescent protein into two halves, and you can fuse the two proteins which you want to test interaction with those two halves. So if both of those complement, then the fluorescence will be there and you can observe it. So we found that some of the NDL1 interacting protein, they do localize into the nucleus, which kind of build up our model that from surface and DL1 move after beta interaction, it moves to the nucleus because beta has been discovered to enter the nucleus. So let's say under normal situation, NDL1 is in the cytoplasm, but when you give salt stress, it will bind to beta and take the message to the nucleus, right? So we found that with one of the two candidates, we found that those interactor of NDL1 were also localizing into the nucleus along with the NDL1. So it's kind of, we are moving ahead. So we selected, these are some of the favorite candidates currently worked upon in the lab. So you, first of all, you confirm their interaction in yeast, and then in yeast also you can perform various kind of growth assays, that if you grow the yeast having this NDL1 and its interacting partner, you grow them in presence of salt. So you can see how yeast growth is taking place, whether they behave well or it's imparting positive effect, then it means that interaction is positive in response to stress, right? So those kind of analysis can easily be, growth assays can be done in yeast. So yeast can be used as a model for those stress assays. And on top of that, you can, the way I said, because there are a lot of mutant available in almost all the gene, you can, and there are banks, seed banks, you can order those mutant directly. So you can order them and then you can just analyze those, whether they are really mutant or not, whether they have that gene knocked out or not at the DNA level. And then you can see whether that RNA is knocked out or not in terms of transcript. And then once you confirm that, you are ready to go ahead with those mutants. You can do phenotypic analysis. I'm showing for one of those that this one of the NDL1 interactor has shorter root compared to col0 and beta mutant. And then you can look for the other phenotype which you expect in response to drought, that you will be predicting that stomata distribution or density or some defect should be there if they are involved in the dehydration stress, right? And we did find for one of those that stomatal density was reduced for one of the mutant. And then you can do detailed characterization with Gus fusion. You can see where do they express, localize. So this strategy can be used for even for the normal basic research, right? When you want to study localization of any protein. So I have taken selected images for various interactor we have worked upon. And then again, the same protein translation of fusion overexpression line, they can be studied on various kind of stresses. You can see how the protein is increasing or decreasing in response to stress. So you are adding to the basic information that how this protein is functioning during the stress response. So with that, like these technique can be generally used for basic research as well as for, let's say, if you're not studying even the abiotic stress, you're studying that one particular protein is involved in fungal infection, fighting fungal infection, the same strategy will be there, right? So you can take mutant of those particular protein, you can take overexpression line. So either you are omitting that protein, you are making more of it, or if you want to study whether two proteins work together, you can omit both of them together, or you can omit one and you can overexpress another in that background. So you can do all those kind of combinations and can study how those two proteins might be interacting at genetic level or how physiologically they are fighting back something, right? So Rhabdops is such a beautiful model that you can do all those assays on plate in the under normal lab situation. And providing stress is also easy. So you can grow them on plate and you can do treatment in liquid or in dishes, right? So can we go a layer down the way I was saying, that metabolome and then biochemical level? Definitely. We know that all these during stress condition, you will have proline or like the sugar content, different kind of sugars, they increase in the plant to fight back, right? So ultimately at the biochemical level, changes are occurring. So we can exploit that property or situation to use some special techniques. So we can use various optical technique also in order to find out what are the differences, or in you can say that you are doing the metabolome, or further down the biochemical profile of the plant is changing. You can detect that using various kind of physics related or the optical technique, right? So let's say plant perceives stress. Now there will be ultimately all these stress, drought, heat, salt, they will lead to the reactive oxygen species generation inside the plant. Okay, and now these reactive species, you have sensor, they will sense those molecule and they will lead to a signaling inside the plant. Plant will know that okay, this particular insult has occurred, now I need to react. And ultimately, the way I was saying for the G protein, the same way, this will go and reactive oxygen species will change the redox potential of the cell milieu, and transcription factor would be affected, and the gene product will be formed, and those protein will be basically for acclimation. They will fight back the stress what plant is facing. So these ROS, or the signaling ROS or the metabolic ROS, they ultimately would affect the different kind of biomolecules which are present in the plant, right? And that would affect the total overall cell milieu, and you can detect that. And these techniques can be helpful at that step. So when you have stress, metabolic changes, and stress specific ROS signature would be there, acclimation or death, whatever could happen, that will be determined. Then let's say, and all these stress signature are specific for specific stress. Stress A would have a specific ROS signature, B would have specific, and both of them together would have a specific signature and acclimation. So various studies has been done, and it has been found that different biomolecules are affected at different level by these reactive oxygen species, and the protein are the most affected one. Okay, and key target of the ROS, and these changes would lead to the changes in the absorption signature of the cell, and which can be optically detected. And I would just give one example that in case of Arabidopsis beta mutant, we have detected that glucose content was significantly high at the root tip, and compared to the wild type plant, which has significantly lower amount. So these kind of changes can easily be detected using various spectral techniques, and terahertz could be one of those. And as it has been shown to be used in the plant for stress monitoring. And I have been collaborating with Aparajita and Amrita on this, and we are trying to find out whether we can do phenotypic profiling of these various mutant and overexpression line using these techniques. And we are aiming to generate biomolecule specific signature in wild type, and then compare it to the loss of function and overexpression line to determine function of these genes in plant. And these genes in long run could be used as ideal candidate for the transgenic approach for crop improvement. And this is just one of the example that is generated by Aparajit in their lab only. And it's not the final experiment, but just to give you a feel that you can generate a signature in the wild type plant, which is normal plant without any gene being defect or knocked out. But then you can do the same signature in a mutant, and you can compare both. So if they have different signature, and like let's say for if the way I was giving the sugar example, and they have different profile, they can be compared, and these signature could be kind of attributed to that gene's function later on. We are in very preliminary stage, but in the long run, we want to establish these signatures specific to various kind of biomolecules. And with that, I'm finishing, and this is acknowledgements. And funding in my lab is from Devi University IV grant, and DST SERV and DBT. And collaborator, most of my beta studies were done in my postdoc lab, LNM Jones. I'm still collaborating with him, and on the interactome part, few experiments are we are doing together. And for the optical techniques we are trying to employ, I'm collaborating with Aparajita and Amrita from IIT Delhi. And majorly the in silico part which I showed today was done by Abhishek Kannochia. during his MPhil. And actually, he was planning wet lab work, but Corona happened, and then we have to move on to in silico analysis, and that's what happened. And Aparna did most of the she's doing implant analysis of these mutants of NDL1 interacting protein. And with that, I would stop, and I would love to take any questions you have.
Hello. Yes. Yes, I think the connection got patchy in between. So I think I was waiting for Yash to finish because I could hear the drag. Okay, so let's thank Yash for a very nice and wonderful talk on the various topics of plants that she has been working on for a lot of time to do genetic modifications in plants and seeing their applicability in different areas and trying to characterize them and do the sequencing of them. And very recently, as she mentioned, we have also been involved in an active collaboration with her and using optical techniques for the first time. So before saying more, let me see the platform is open for questions. And so if you have any questions, please feel free to unmute yourself and ask the speaker.
I'll have a question. Thank you for a very nice talk. Very interesting to see your work. I was very impressed and interested to see how you can change the plants to be able to survive different kinds of stress. I was curious what you can see about variations in nutritional value, for instance, for surviving drought or salinity like you showed. Can we also get gains in the nutritional value of the crops that way as well?
In response to stress?
Yeah, well, so if the plant is surviving under drought or under different salinity conditions, as you showed, I'm curious about if that has any impacts on their nutritional value at the same time.
Definitely, because they do reallocation of the biomolecules, because if a plant is sensing stress, now it needs to fight back, right? So it will just have the essential processes going on, and it will avoid any loss of energy. So it would rather kind of try to survive and fight back instead of making, let's say, more seeds or more yield. So normally under normal situation, most of the yield traits are going down. But in case of halophiles, their plants are happily growing and they have adapted to the environment and they know how to survive in the stress situation, like agave and the other plants. They are not affected. Their their nutrition value still be intact. But under normal, I would say normal crops, they are adversely affected.
Thank you.
Welcome.
Okay, thank you, Yash. And do we have any more questions? Okay, if not, let's thank the speaker once more for a very wonderful talk, especially about the stresses and all the other various activities that she has been taking over the course of her research career. So with this, we will now move on to the next speaker of the evening over here and of the morning in Canada. And the next speaker is Sudeshna Chattopadhyay from IIT Indore. And she will be telling, she has been a faculty at over there and has also had a visiting faculty position at the Department of Physics at RWTH Aachen in Germany. And her research interests include surface and interface science, synthesis and interface engineering of advanced materials for energy conversion and electron.
Oh, Amartya, you became muted by accident. Sorry.
Yeah.
Okay, so let me just go back once more. Yeah, so the Sudeshna research interests are mainly advanced materials for energy conversion and electrochemical energy storage systems, yield deposition, and soft matter physics. So her topic of the for the evening over here is effect of interface engineering, rechargeable thin film batteries, And so over to you, Sudeshna. Just one point. You have 25 minutes for the presentation and 5 minutes for the Q&A. So just to indicate that your time is, you have about 5 minutes left, we will turn on our videos. So as of now, all of us will turn our videos off, the organizers, all three of us, Amina, Aparajita, and myself. But when you have about 20 minutes into your talk, we'll turn it on. Sure. So that you have an ending. Okay. Thank you very much. So please get started. Thank you.
Okay. First of all, thanks to Dr. Amartya and also all the organizers, that is committee members of the organization committee. So for inviting me to this to deliver the lectures in this conference under the umbrella of Indo-Canadian bilateral cooperation program. So I'd like to thanks and say, now I just wanted to go to my presentation directly without spending more time on this. So I think you can see my presentation. Could you please confirm me?
Yes, we can.
Yes, we can.
Yeah, sure. So yeah. So now, as I just wanted to tell that already, we have just in the morning, we have heard from the Google India from Dr. Alok Talekar, right? So from him, that how the energy research and energy storage system, research in the energy storage system are very much connected with the climate change, and also, of course, the many other issues related to the climate change and environmental remediation kind of. So so I don't want to give this kind of introduction anymore, but just I am going towards the direct some topic on the energy research. So of course, as my title already mentioned, that effect of interface engineering in energy research, and the kind of case study for a rechargeable thin film batteries. Okay. See here, what we just wanted to make, point it out that, okay, because whenever we are working on the energy storage or energy research, of course, in chemistry point of view, we can develop new material, new system for that. On the another way, for the physics background people, that it will be very, very important and of course effective as well, that if we can just for a given material, if we change the interface, that is interface or surface, because interface is nothing but surface is nothing but an interface of air and the system, or vacuum and the system. So surface interface modification can provide you the even more better performance for a given system, which is in normal situation, in its bulk state, it is kind of inactive for towards some applications. So how can you do that, and why it will happen? So that kind of important information just I wanted to share with you with some examples. Okay, so that is just first I just wanted to say about our Canadian and other international people, that okay, Indore is located at the center of India. It is very close to Delhi and also from the Mumbai. So we have the good collaboration with Dr. Amartya also. So if you visit, you are most welcome to visit at Indore also. So my research area in this related the surface and interface science, as I have told, because I wanted to give some glimpse on that, because soft matter physics and excess scattering, electrical energy storage, heterojunction, hybrid material, these all whatever I am saying here, these are again connected with the application of how can you modify the interface and surface, and how can you get the important information in terms of basic science and in towards its application. Okay, that is the goal. And whenever I am talking about surface and interface, it will go towards the nanoscience or nanostructure. Because why? Because surface will be important only when the the material size will be very small. Very small is a relative term. So if we make it a very particle size is such a way, if we confine that, so that surface to volume ratio, that means surface area to volume ratio will be higher. That means surface molecule will dominate, or surface atoms will dominate in the to play the role to determine the property of the system, then only that effect we will be able to see. Otherwise, in bulk material also it is there, but we would not be able to see overall effect, because bulk number of molecules or number of atoms within the bulk, it is so many with respect to the surface atoms. So we that will not at all affect the whole properties of the system. That is why when I am talking about this interface and surface things, it will be mostly useful when the system will be very small. Either it is nanodots, nanoparticles, or thin film that is in scale of nanometer. The nanometer, why we are saying it is because then surface will that effect of surface you can see there. Okay, so for this work, of course, many funding agencies and all from my team, though there are lots of supports. So I'll just go towards our main focus, the surface and interface. As I had told, soft matter physics already Dr. Amrita has mentioned, and of course biomedical applications in nanotechnology for the environmental remediation, also different field, and of course the energy research. Today I will talk about that. So targeted problem is the design of novel materials for energy research or nanoscale device in the field of flexible electronics, photonics kind of effect, and then medical diagnosis or environmental remediation related problems. Okay, so some selected publications related to this topic from our group and from my research field. Okay, and then I just wanted to focus today energy research program. So of course we know that ever increasing global power requirement, which you cannot tell that today, okay, I am satisfied whatever we have. No, it will be always you need more, because this energy requirement, because our application is going every day, our expectation is going high because of our different application in the electronics and every field. I would say agriculture, every field we need more energy. Every day we'll feel that okay, need more. So for that we need high performance advanced material technologies for alternative and sustainable energy. And now if I go for this system, so we have to focus on two major things. One, energy conversion, where you will get the energy. Of course, finally solar system is the we have to get the energy conversion from the solar system, so that will be the ultimate objective, that means for the to get the energy. And second one is the storage. So storage is also important, because if we can't store, so then we cannot use it when we need that, when we cannot get it directly, the solar energy conversion system active based on the situation, maybe in the evening, maybe during the requirement process. We need the grid or energy storage system for that purpose. So, but major challenges are there. Of course, we are working. We have the electrical energy storage or electrochemical energy storage. Sometimes you can tell. So there are different kind of energy storage. I will focus on the battery, which is called the electrical energy storage. Sometimes electrochemical energy storage. So in that case, major issue is that we don't have the high performance. High again relative term, because whatever our requirement, we need more performance. So safety issue is always there, because lithium ion battery is the most commonly used battery system energy storage in energy storage field. So but there is a safety issue is major. We can't avoid that. And then again the lifetime, better cycling, stability, and finally the cost and capacity. So because lithium ion battery, the whole system, when you were working in the car or anywhere, so it is very expensive, all of us know. So electrical vehicle, the main cost goes with that. So then the search for the efficient electrode materials for electrical energy storage, I'll focus on that. And if I get some time, so I'll give some glimpse of our work related to the just again the surface interface effect. How can we get the conversion of the energy, just only changing that, okay, for few nanometer, few layer of interface with the new molecular interface modification, how you can get the abrupt change in the application. So that I'll discuss. So then some example of surface interface affectation. It is not only the energy storage. So there is a particle, you can just get the form. So how can the water in merges with the system, that those kind of many effects are there. So here, since, as I have told, I'll just go for the battery before that energy storage basic things, since we have the different audiences from the different fields. I just wanted to quickly say about that battery, how it works, that very schematically, very crudely. So this is the electrode 2, and then within that there is electrolyte. All of us know that in a 7th, 8th grade we have learned that. So after that, what we just important things we have to be aware of that. So that electron conduction and ion conduction, because electrode should that for the electrode systems, of course we should have the electron conduction in the electrode system, but that electrolyte, in between there will be an electrolyte which will help you to just separate these two electrodes to avoid the safety, that means safety related issues, so that it will not be short connected. That means two electrodes will not be connected. Okay, so that is the separator. So that should not be the electron. Electron should not conduct through this. Okay, that is the important feature. Second feature that we can predict. Okay, that should be there. Second part is that ion conduction. So ion conduction in the anode and cathode, it can be, it may not, that means, I mean, it is not must requirement, it can be. Okay, but on the other hand, the separator should have the ion conduction. So that is an important point. Okay, so we should remember that the battery part. Okay, so now that if we just wanted to, as I have told, electrochemical storage or electrical storage, what you can tell that. So capacitor and batteries both are within that field. Okay, so but what is the major thing? So when we are using when we are talking about the capacitor and batteries, maybe during my work I will just tell a little bit about that. So because if you see that in x-axis and y-axis, there is a specific energy, that is a specific power. So for capacitor, we are getting higher power density, or specific power is higher, and on the other hand, batteries used to show the higher specific energy. So in this x-axis, so we are trying to get something in between also. Supercapacitor, people used to work on that as well. So where you want to get the higher capacity and higher specific energy and higher power. So our goal actually is to get some storage system which will have higher specific energy density and higher specific power density as well. So that is the goal. So we want to reach there. So somewhere like this corner we want to go. So this x-axis will be high and y-axis will be high, and high is again that your scale will be getting increased with time. Okay, so your requirement will be higher, but we want that both. So then for a good battery, what you need, that of course, as I have told, high energy density we used to get in general. So of course it would be much better with respect to the whatever we get now. Second one is the power density, rechargeability, lifetime, safety, electrochemical stability, thermal stability, low toxicity, and wide operating range. That means wide temperature range it will work, and like that, cost effectiveness. So rechargeable lithium ion batteries is based on their high energy and power density. Of course, other things are there. So it is at this point widely used in important equipment. Okay, but the problem is still there, because there is always an issue arising from their combustible organic liquid electrolyte, because these such kind of issues will be more prominent when you will use it in a bigger aspect. That means I'm saying that for the EVs or like that grid system, if you want to use that kind of LIB, so that will be more prominent, because whenever that effect of the organic liquid electrolyte will be, amount will be higher and it will be affected more. So that is the major issue. So now, of course, we in our group also we are working on the LIAPs in different ways. So how we can make it better. Of another many other research groups still working on the lithium-ion battery. But in addition, we are working also on the batteries beyond the lithium-ion. Okay, so which is aluminium-ion battery specifically. Of course, there is a zinc-ion, potassium-ion battery. People are working on that, but we have selectively chosen that because of some the inbuilt, I mean, advantages of aluminium, which is most abundant metal in the world. So it is kind of low cost in that respect. Okay, much low cost with respect to lithium, and it is, you can get it huge amount. There is no deficiency in terms of the availability, and it is safe, so you don't need to think about such kind of safety issue for the lithium. Okay, and then it has a high gravimetric and volumetric energy density, low flammability, and of course the eco-friendly non-toxic materials. So, but of course there are some issues. So if it is everything is good, so you could have done everything. So the issues are major things, the capacitive behavior without discharge voltage plateau. So why I was saying that capacitive behavior means, you will see that afterwards I'll show the charging discharging features of the battery. So you will see that if it is a capacitive behavior, so ions will go on the electrode and it will stand, so it will just adhere there. So it doesn't need to go intercalate to the electrode system. But in the case of the capacitor, it can adhere and it can go back and forth like that way. So when the ion will intercalate to the system, so it will show a charge plateau for a certain voltage. It will show a flatter system, and that will provide you the impression, find that this is the battery type behavior. So for aluminum and battery, since the very good electrode system we don't have, so that means we don't have miss at this point. So that is why that most of the electrode system doesn't allow it to go inside. So that is why capacitive behavior we used to get. And of course the open circuit voltage is also low because of the electronegativity, the amount, how to calculate the open circuit voltage based on that. And the capacity and cycle life are not satisfactory at present. Okay, that is because I have shown some kind of examples. So good journal paper, the references are really good, but this is very low. I mean, previously whatever it has been. If you see the most of the paper for aluminum and battery, it is 2012. 11 after that. Okay, because the major problem was that people have understood theoretically it has a very good capacity. That is why people were interested to work on that, but the suitable electrolyte was not available to to conduct this aluminum ion in the battery system. So when chemist has developed that electrolyte, appropriate electrolyte, around I think 2009 or so. So it was established for the aqueous and the aqueous and non-aqueous electrolytes. So after that, research on aluminum battery got really accelerated, and you can see now the many publications, patents are coming on that. Okay. So I'm just not going to the thorough calculation. It is not a brief thing. So I just wanted to mention that now what is the point of the thin film, because I have told the thin film battery. So aluminum ion battery and thin film battery. So thin film battery, why we are talking about? Because low film thickness of electrolyte will help you to get the better ion conductivity. On the other hand, low film thickness in the electrodes, so you will get the better diffusion rate. So which plays significant role in enhancing the power density and high rate capability of the battery. So which we want to get it. So that is why thin film battery will be a kind of useful for, of course, for some applications. So not always applicable for all the systems, but it will, for some application, it will be really good for this kind of advantages. Okay, so we had focused in this works, whatever I am going to present here. So it is aluminum ion battery, thin film battery, but electrolyte is normal electrolyte, just liquid electrolyte. It is not film, that means normal electrolyte, but we have just use the very thin film electrode system, okay, that cathode system, because anode will be the aluminum in this case, and the cathode in the because standard, so that we can see that what is the effect of thin film cathode. So thin film cathode, so here we have selected the zinc oxide. So why it is zinc oxide? Because it shows a very high theoretical capacity. If you estimate the theoretical capacity of zinc oxide in presence of aluminum, it will be kind of 2900 milli around, I am saying that milliampere hour per gram if we calculate theoretically, okay. So that is, I just wanted to give a comparison. If you just see that nowadays for the normal very commonly used lithium-ion batteries with graphite anode, so in that case we generally used to get 370 milliampere hour per gram, which is very, you know, stable, and of course recyclability is very good, other things are very good, but capacity wise this is around that. So in that case, that respect, if we can achieve 2900 or even half of this, that will be also good with with such a low cost material and safety issues we can resolve. So that way we have selected the zinc oxide thin film electrode for such system. So now we have used the atomic layer deposition technique to deposit it. Why? Because whenever we are talking about very thin film, so we want to make the system such a way so that it will provide you the, because at least objective is like that way. So atom by atom, when it in a atomic layer deposition, it is just kind of working principle we have shown here. So it is kind of layer by layer it used to form, but so we can control very clearly the film thickness, even 3-4 nanometer to the, you know, higher thickness as well. So the important point is that with this deposition technique, we can expect that it is pinhole free kind of deposition. Because why? We don't want to be exposed, that is the current collector should not be exposed, okay. So with the electrolyte, that is why we want to get that kind of pinhole free deposition. That's why we have selected that method, and we have used this for this purpose, okay, in our lab. So now, before using that, we have to see how the morphology is there, what thickness we will choose exactly, because we can deposit many different thickness, as I have discussed. So of course we have to optimize that thing. So within that we can see that. So if you that X-ray reflectivity, we have that equipment and also analysis procedure for the expertise on that. Because why we need that? Because whenever we are talking about, as I have told, our work is surface interface effect and nanostructure. So if we form something, I can claim that we have formed, but how can I measure the thickness? Because that is very crucial. Because what is the thickness? So X-ray reflectivity provides a very good estimation for the film thickness in Armstrong order resolution. So you can see that these are the Key-Seq fringes, and we can extract the data for the zinc oxide and after that silicon, because we have chosen first the standard sample to optimize the thickness, and we can see the morphology, how it is changing from higher thickness to lower thickness. So we can optimize the film thickness whatever we need for this battery application, okay. On the other hand, from XRD pattern, we can see that zinc oxide 002 peak is much stronger with respect to the other peaks, so which indicates that it is a textured sample. So textured is also here. So now we'll see that how we can utilize this system for our application purpose, okay. So here we have done some other electronic property check also with ellipsometry on the dielectric function and like that, because that was an important requirement, because as I have shown in my initial slides that electrode should be electron conductive, right? So that we need to check that conductivity, how it is, and with those things of course connected with the electronic property. So electronic property and then again optical properties, these are correlated, dielectric function and like that. So from there, thorough calculation, and we can just estimate that, and of course from the photoluminescence we can get the band gap and defects levels of the system, those things. So we have to standardize those things. And of course, we have seen also important part from there that, okay, interface is very important in that case. So if I deposit zinc oxide on silicon, and if I deposit on silicon oxide, and if I deposit on other material, so it would not behave exactly same way whenever it is very thin. So if it is very thick, so it will kind of standard, so interface will not be dominated, okay, as I have mentioned earlier. But whenever it is thin, I have to take care of the interface as well. So what is happening in the interface layer, we have to be careful on that. So because of that, we have checked that from the lithometry analysis, so we can see that how interface is affecting on the affected by the substrate, and based on this, we have taken the precaution when we are depositing the zinc oxide on the steel, because for the battery application, because for the battery application, I need the current collector on the electrode system. So current collector, generally people used to use the steel for this, electron conduction. It helps the electron conduction from this system to the cathode to the current collector to that is why. So that way we have chosen this. Okay, now next step after standardization of the different, so what we'll do, so we have just deposited the films on the steel around 20 nanometer, 20 to 50 nanometer, different thickness. We have just cross-checked that which will be the optimum, but within that range I am telling. So you can imagine that it is very thin. I have estimated the film thickness also from the ellipsometry as well as from the actually on the steel we have estimated from the ellipsometry, because steel roughness is very comparatively within the micron order. So for micron order roughness, we cannot get the X-ray reflectivity data, but ellipsometry is very good to get this results. So film thickness we have checked from there. And then we have seen that from X-ray diffraction, so it is the textured material. So we have you have shown schematically. Of course, this is the ideal situation. So zinc oxide looks like it is aluminum cathode. It is a separator, standard separator for aluminum and battery. And then you can see the effect. That is a deposition of the LD grown zinc oxide here, okay, that which is textured we have shown. So then photoluminations, why I am showing this is showing very nice defects, because defects we generally try to avoid, but sometimes defects is very important. Because why? Because defects will give you the ion conductivity, electron conductivity better in that system. That's it will behave like a doping, so like that way. So here that is also important. And for morphology, you can see there is phenol-free kind of system, of course, that for an example. Now the important interesting point here, if you see that the discharge capacity for our system for this 20 nanometer to 50 nanometer, as I have told, that film cathode, so it is showing the capacity around for the first cycle, of course, 2500 around like that way, okay, milliampere hour per gram. As I have told, 370 on an average of the lithium battery. It is really good. Of course, charge capacity is less. So I will show the Coulombic efficiency afterwards. So this model, and you can see that this is the perfect coin cell making, and after that it is just completely, we have checked with the LED also, because we have connected 3 battery here, not to get the more kind of current, but it is because if you see the open circuit voltage for aluminum and battery, it is around 0.8 volt, but to lit up that red LED we need at least 2.1. So that's why we have connected this 3 to get that. It is working perfectly, and whatever in time is also good, but you can imagine that is only 20 nanometer cathode. Okay, so then now this slide, so I just wanted to show that, of course, it is fast capacity is really impressive, and after that capacity is not, it is going down, but you can see that 8 cycle, but still you can see if it is higher. Of course, we need to, that is capacity 800 milliampere hour per gram, like that way. And as I had told earlier, the charge plateau, charge and discharge plateau in these things also, you can see this is the plateau, okay. So this indicates that yes, it is not the capacitive in nature, ions are going inside the system and getting out. So intercalation and deintercalation is prominent which has been confirmed again from the CV data. So you can see the reduction and X-oxidation peak is consistent, stability is high, but of course Coulombic efficiency is also very good. But of course the capacity for the initial cycles is getting down afterwards. So for the betterment, if we can avoid this down part, that means some way we can avoid some ACI formation. We are thinking that okay, when it is forming some ACI formation means solid electrolyte interface formation, that is maybe restricting that efficiency capacity. That is why it is getting down. But still, when it is standard, then it is even 300. So I mean saturated after a long cycle. So it is still good with respect to the available battery system and like that. Of course, if we can get it higher, because why I'm saying it is safer, it is much low cost, and it is very lighter because amount is very less. Okay, so then we have done the diffusion and the coefficient. So it is perfectly, it is showing from the PL, we can show the changes and like that. Photoluminescence, XPS data, it is clearly showing that it is going inside, because whenever we are talking about a new system, we have to make sure that okay, it is really happening this way. So you have characterized. But important point I wanted to note, inform here, that that we have checked that why the zinc oxide, any other system is not good for that. So we have checked with the nanoparticle, just commercially available nanoparticle, how people used to do that for the battery system. So the slurry, making a slurry with the conducting material and also the binder, and then we have done in the coin cell, the experiment has been done, and we have seen that nice, that is the very less capacity for that zinc oxide bulk and nano, but it is for ALD grown ZNO, that is very good. So why ALD grown ZNO? This is a summary there. So because we can we can just mention from the characterization, it is showing high capacity. So what is the reason? Possibly it is giving it because of the textured C-axis oriented ZNO growth within that system. It is really facilitating the good electrical contact and enhanced pathway for electron and ion transfer or transport kinetics, which has been also enhanced by the diffusion of the for the defects level generated by the zinc oxide growth system because of the surface. Because surface, whenever you are making that, oxygen deficiency will be there, and because of the very thin film, so oxygen deficiency will be prominent. That is generating some defects levels in the system, and it is giving you the more conducting property. So we had to use some DFT calculation with our collaborator. So then it is showing that it is giving the favorable energy system for the aluminum ion intercalation phenomena within the framework of the C-axis oriented zinc oxide. So that is the major things, and quickly I can just tell you that another aspect of that 1 minute or 2 minutes within that. So another aspect of the system of the surface interface coating, where you can just convert the solar energy system. So we have shown for the electron hole pairs, all of us know that solar system, whoever are working. So for the silicon carbide system, if we graphitize that system, only 1 or 2 layers of graphene, how it can enhance the your capacity, I mean for photocatalytic activity. So we have shown that for normal ideal silicon
Sudeshna,
carbide,
sorry, can you please try to wrap it up?
Yes, I'm just going.
Thank you.
It is completed. Yeah.
Thank you.
So that is the things, and then we can mention that okay, this is again the 1 or 2 layers of graphene, how it will be useful to make the system very effective. So again, this example is to show the effect of surface and interface in energy research to convert the band gap narrowing around 30% and enhancement of the capacity even 1000%. It is so that way we can just emphasize on the system, and then I will acknowledge my research fundings, and for the of course SCRB and CSIR and DAD still we have the, some mobility on this program and other research fundings from different international and national agency and IIT Indore, and then my research group, and then also my collaborators. So there are many other collaborators I just, who are with whom we are working regularly generally, so that I just mentioned some. And this is that's it. Thank you for your patience, and thanks. Yeah. Have any question?
Okay. Thank you, Sudeshna, for this wonderful talk. The platform is now open for questions. So participants who are interested in asking questions to the speaker can please unmute themselves. We have time for about 2 questions. Okay, so if there are no questions, or while other participants are thinking about, I have one question, so maybe I'll just put it forward. Yeah. So what is the prospects you think of these sort of batteries that you're working on? Or for example, this EV technologies and all these high capacity batteries that you're talking about to come into mainstream applications in, for example, let's say, if somebody is trying to develop some photonic sensors, battery operated photonic sensors to go into space, for example, for some planetary explorations or for some drone applications. So what do you think are the sort of prospects of these things?
Yeah, I think that would be because that is why just when I am talking about thin film battery, so that is really, really applicable for such kind of application, because when, majorly, of course, in other applications is important, but such kind of applications, it is essential that we need to have the light battery in terms of weight, as well as it should be very thin film, because otherwise it will, space or drone, whenever we are talking about, we need to have a very light material, as well as we need the high capacity. So if we can give it, because kind of as you have understood maybe within that if we can make an aerial only kind of coating, some examples I'm telling, so coating of this kind of system, okay, so with the solid electrolyte, because nowadays working on the solid electrolyte is the one aspect, so I have just started on that, as I have told, electrode system, and next one is the solid electrolyte. So if we can make it this and then we can coat, so it would be really useful for such kind of application, because we have seen that okay, it can just work LED and like that way, but after that if we just make it in the proper way, and because any kind of, if you just think about that, it will be coated on the surface of the drone, okay, so that will be useful in that way to get it convert, energy storage as a system. You are not using additional part, okay, I'm saying that way. So that is the objective for such kind of research, in reality.
Okay, thank you. Okay, Aparajita has another question.
Yeah, I was just asking Sudeshna that since you very wonderfully put that how you envision that without actually increasing the payload, you can still use the drone surface itself as a battery. That is actually very wonderful. But other than that, I mean, the usual concept of battery, the traditional, basically just a block of this kind of thing. So is there any chance, or is there any rather efforts from the energy industry that to basically sort of recycle those things, maybe not with full capacity, even full efficiency, but maybe in the maybe industry's floor or somewhere where it is not you are not going for mileage or distance, but rather very small, because even we know that even in the sort of the factory floor there are so many goings and comings, and so can we do certain things, or is there any efforts regarding that?
You mean that recycling part,
exactly, right? Recycling the already EVs that are being produced in a more traditional fashion, like the lithium ion batteries I mean, because their efficiency goes down pretty quickly after certain cycles, right?
Yes, yes, yeah. So in that case, we have shown maybe in the some slides, so you can see that. So we used to do that. Many cycling is very important. I mean, life cycle should be much higher. I'm saying that we have shown that the 100 cycle, of course, that for this, and it is kind of saturated after some time, because the objective is, if you see that one of my graph, I was saying that, okay, capacity is much higher. Initial first 2, 3, even 15 cycle it is really, really impressive, but it is going down, of course, in the experimental pattern. But our objective, but after 100 cycle, even if it is around 300, so it is not less with respect to the available battery. So that is the things. But it would be really good if we can enhance this, that after first cycle, whatever we are getting, when it is dropping down, so there is some reason, right? So we have predicted some reason that, okay, maybe some say ACI formation is there. So maybe some coating or some interface things, so which can stop such kind of drop down the capacity. So then it will be much better, of course. But at this point, if we consider, okay, first 4, 5 cycle, it will give a good amount. And after that, we will take it as a whatever is available, that amount, but we are gaining in terms of cost and safety, because aluminium is safer, cost wise is less, and also that another one is point is that lighter, because it is we are using very, very thin film, as a 20 nanometer, you cannot, kind of weight wise is also the thing. So these 3 things we can gain, but to get the equivalent things after maybe 50 cycles or so. But initially it is much higher. But if we can get it completely stable, so that will be the kind of goal to reach there also. Yeah. Yes.
Wish your team all the best.
Yeah,
Go
thank
there.
you. Thanks.
Thank you.
Okay, thanks.
Okay, so we are running a little bit back on time, so we'll just cut short the interaction. Thank you for your understanding, and thank you once again, Sudeshna, for the wonderful talk and the insight that you gave on electric batteries and the possibilities of electric energy. So with that, we'll move quickly to the next speaker. I think the next one will be a very interesting talk, as have been the previous ones. It will be given by Logiag, a company. And the Logia company is essentially a team of agronomists, technicians, and computer programmers with over 10 years of experience in the agricultural field. They are based in Quebec and have recently been awarded the Indigo Carbon Challenge Award for finding novel solutions in agricultural soil carbon sequestration using the laser technology, which is based on LIBS, or laser-induced breakdown spectroscopy. So today we are fortunate to have 2 speakers from them. One is Jacques Noir. I hope I pronounced the name correctly.
I think Amartya might have cut out. Am I the only one for whom that happened?
Kind of.
No, I can't hear him.
Yeah.
Okay. Okay. So I can continue the introduction. He's back. Okay. Yeah. Looks like he's back. Sorry, Amartya.
Can you hear me?
We can hear you now. Sorry, you cut out
just
after telling us it's based on libs. You can continue.
Oh, okay. Okay, so now sorry about that. I think the connection is patchy. Okay, so we are fortunate to have 2 speakers from there today. One is Jacques Noir, and pardon me if I pronounced it wrong. He is the vice president of the company and has 30 years of experience as an agronomist. And he will be joined by Dr. Christian Gugel. Again, pardon if my pronunciation is wrong, who is the director of research and development for Logiag. And so what essentially is that Dr. Noalt will first do the introduction by presenting who they are and the role laser can do by accurately and efficiently measuring soil organic carbon. And then Dr. Gugel will follow that up with discussions of scientific and technical aspects of the technology. So thank you very much for agreeing to join in this forum. And we're looking forward very much to your talk. Thank you.
Thank you very much. Thank you for inviting us. Like you said, we will split the presentation, and I think Christian will share his screen so that we don't go from one screen to the next. So Christian, maybe you could put the presentation on, and I will start speaking. Is Christian there? I saw him before. Amina, can you see him?
Yes, I can see him there, and it looks like he's a co-host to share. So I'll just send him a chat and make sure. Yeah.
Yeah, I just want to because I'm going to start and he will continue, so he will control the presentation. We thought it would be simpler this way.
Perfect. That sounds good. I've sent him a message as well. Okay.
Yeah. Christian, you are there?
Yeah, it's okay. I just send you the presentation so you can present because with Zoom, I'm using Mac, and Mac and Zoom they don't work good together. So I just send you the presentation on your mailbox so you can share it with everybody. It's okay.
So I'm sorry, Christian, you want me to share it, to share my screen? Is that what you're saying?
Yeah, because I have some issue with my my computer right now.
Okay, so let me share my screen then. Sorry about that. Can you see my screen?
Yes, we can see it now. Thank you.
Yes, we can see it.
Okay, very good. And I will try to put it this way. Okay, so what we're going to share with you today is we're going to talk about laser ag, which is an adaptation of the laser-induced breakdown spectroscopy that my company and my team developed over the last 10 years with some collaboration. I will talk about this in a minute. For agricultural purposes, and we are now able to use it to predict nutrient content of soil for agricultural purposes and plant tissue analyses. And recently we won the Indigo Ag Carbon Challenge in the quantified track, showing that we can also analyze or predict correctly soil bore density, soil organic carbon, soil organic matter, and a few other parameters. So I will speak, the first part is I'm the owner of the company. I will talk about the business side, and Christian is the scientific director. You will talk more about the scientific technical part of the technology. So a little outline, I'll say a few words about Le GIAG, then I'll present the instrument, which we call the Laser Ag Quantum, and Christian will talk about the determination of soil texture and the measurement of soil organic carbon with some results, and explain how the technology works. So first, a word on Le GIAG. I founded it in 1999. And we are currently, I say it very with humility, we're a leading agronomy service provider in Quebec. We develop all our own software and agronomic solution. And we've been working on the laser ag system. Like I said, it's an adaptation of the LIBS technology for agricultural purposes. And our partners in the research were the National Research Council of Canada, which originally, this is where the Canadian knowledge on LIBS was located really, and we started to work with them and to adapt it for our purposes. And we also work with l'Institut National d'Optique, which is a leading institute in optics, and they are the designer of the commercial instrument, and they are also our manufacturer. And like I mentioned just a minute ago, we were in the Indigo Ag Carbon Challenge in the quantified track, and we won the challenge. Basically, we showed that laser ag was very efficient, very accurate to evaluate or predict stock of soil organic carbon within the challenge that Indigo had put. And it was an international competition. There was 230 other competitors originally, so we're very proud of this. I should have pressed that button. This is the building where most of the time we work. Since the pandemic, most of our employees are working from home, but I'm at the Laser Ag Quantum Analyzer. So the instrument, this is what it looks like. We called it the Laser Ag Quantum. Some of the advantages and really the characteristic and what makes it such an appealing technology is that it takes only a minute to make an analysis and to make a complete analysis. When you put a sample underneath the laser, it will take a minute before, and less than a minute, to get the complete soil or foliar analysis. When I say complete, it will include soil texture, soil boil density, soil organic carbon, soil organic matter, pH, level of pK, calcium, magnesium, micronutrients, and so on. It's very efficient, and currently our level of accuracy resemble those of more conventional chemical extraction for most parameters, not for all parameters, but for most parameters. We achieve similar results or ability or accuracy. And of course, we're extremely efficient and extremely accurate with soil organic carbon and organic matter, boil density, and texture. And Christian will explain more about texture and the soil organic carbon in a minute. This is what a spectral image, so the instrument capture an image, a spectral image. You have on the axis the wavelength. Christian will explain more how the technology works, but basically we intercept photons that are excited because of the high temperature. We measure wavelength. So the axis here is the wavelength, and the y-axis is the intensity. And we've developed, like I said, our own software, and the system comes with a user-friendly interface to facilitate the interaction with the machine. The way it was designed is we put a carousel inside the instrument. The carousel has 14 spots. So we put 14 samples. We turn it on, and it goes, the laser goes around the carousel, analyzing the 14 samples. So it takes about a little bit less than 14 minutes to get the sample, all those samples analyzed. This is a typical Lib spectrum on soil. The range is 200 to 820 nanometer range. And when we shoot the laser, we basically see almost a whole periodic table every time. So the real difficulty in this system was to be able to eliminate the background noise and concentrate on the elements that we were interested in. But getting the results on, for example, on magnesium or on calcium is absolutely useless in agriculture if you are not able to translate the information in available nutrient or exchangeable nutrients. So, or pH, for example, or organic matter. There's no reason to get carbon if you cannot get organic carbon, or if you cannot get soil organic matter. So the real genius of our system is in the algorithms that translate the spectral image that you see on the screen into usable agricultural information that can be used to produce nutrient management plans or fertilization program or management program for farmers and for agronomists. And that is what we have been able to achieve. So laser ag is fast, fluid, accurate, and green. I say green in the sense that it's environmentally friendly. There's at the end of the process, all you have is a recyclable plastic cup and soil. There's no material, there's no chemical produce. It uses very little energy. You don't need a special energy source. It generates no greenhouse gas. That's why we consider it as a clean tech as well as an ag tech. We've also developed, of course, we're in the agricultural field. We serve 6,000 farms. We cover 2 million acres. We've developed a software for sampling aid. So on the left of the screen, we've developed a software that we call a LaserAct Partner, and you use it to import shape files, to draw, feel, contour, and to plan a sampling pattern. Once this is done, it synchronizes with a mobile app, which is on a smartphone. And the person who will actually go out in the field and sample, once it's near the field, turns on the app, and it will guide it to the various points that the supervisor has intended to be sampled. And each sampling point will be geopositioned, and the sampling app will also have, if you see on the screen, there's a little icon for, this icon here is for a QR code. So it registers a QR code which make identify the sample with its position, but also with the field and with the farm identity. So once the sample get to the system, the instrument for analysis, you don't need to fill out any forms. It's tracked throughout the process up until the recommendations. The way we see ourselves within a business environment is that we use a lot of information to make a prescription or recommendations for farmers. The information and the data comes, of course, from the farm, but also from the machinery of the farm. For example, the yield monitor on combines will give us yield information, seeding information, and so on. We get weather information. We can get other source of data on soil or anything that's available. And of course, LaserAct Quantum is a system that can provide efficiently soil data into a software system that allows agronomists to make prescription. And of course, now we're very much interested in the climate change, the role that agriculture can do in fighting climate change, and in the carbon sequestration in soil. And so we're developing a system that uses the soil information to follow the tendency or how farmers' practices affect soil organic carbon, and hopefully to keep track of carbon credit, or at least keep track of the carbon footprint of the farms. But this is the model in which we find ourselves, and we believe that LaserAct Quantum and the LaserAct system has a special place as a source of soil data to feed platforms or a software system and help make better decisions. And at this point, I will pass this on to Christian, who will explain the more scientific aspect of things. So I will unshare. So Christian, I think you are already there.
Yeah, so
okay.
I will go. Okay, I will go follow with the presentation for. Okay, just here. Sorry for that. Okay. So I will present two different applications. The first one will be how to determine the texture of soil samples, and the second one will be to measure actually soil organic carbon.
Sorry,
And,
sorry to interrupt. If you could maybe just hit swap displays on the top, because we're seeing the smaller version of the slides on our screen.
Oh, okay, sorry.
No problem. Thank you very much. It's just in the top left corner next to end show.
Okay.
That's perfect. Thank you.
Thank you. So for the first application, so determination of the salts, the texture. So actually, the conventional method, you have three usually common method used in testing lab. It's the saline method and two sedimentation methods using pipette or hydrometer. And those methods are standard methods. They are accurate actually for the pipette and hydrometer. And but the most drawback of that method, they are very laborious. And if you have, you can think of if you have thousands of samples and you want to have the texture of the salt, so you can see that it takes a lot of time to do those measurements for each individual salt, and also they are very expensive. So and that's why certain farmers, they don't actually measure it. So they prefer to measure more nutrient, but the salt texture, it's a little bit left a little bit. So just for the cost of the measurement. And the goal for the laser technology was to try to use the laser technology to predict the texture of salt. So when we talk about the texture of salt, so we need to accurately predict the value of the amount of sand and silt and clay. Those are the three mineral particles. So we need to have an estimation of the texture of the salt. So for the leaves, because leaves is usually used for determination of minerals and different chemical elements. But in terms of the salt texture, we want actually to measure the physical parameter of the samples, and for that, fortunately, the leaves, we can see that in the two graphs on the left, on my left. So we have how the leaf signal vary with the texture of the salts. We can see that the leaf signal have those information. So there is a perturbation of the texture to the intensity of the leaf signal. And on the right, so I just put an example of the PCA modeling for I think maybe hundreds of soil samples. We can see that actually we have two different parts of cluster. When you see the green one, it's mostly sandy soils, and the blue one, it's clay soils. And just in the middle, you have a mix of silty soils, loamy soils. But this gave us an indication that, okay, we can use the technology to actually predict like the texture of the soil. So in terms of, so we have used different chemometrics algorithm for that. So PLS, ElasticNet, and different ones, mostly linear algorithm. So we can see that for the clay, silt, and sand, we have a good prediction for those elements. You can see that the amount of the accuracy increased with the from sand to clay, and this it's not. In fact, it's really related to the size of the particle by itself. So we're going to be more accurate with very fine particle size than large particle size. So there is like a tuning of the experimental parameter to be done just to increase those accuracy. But just quickly, you can see that okay, we have like a good capacity to actually predict the soil, the texture of the soil. And I will just quickly move to the second application for it's the measurement of COC. So for the soil organic carbon, so here we have maybe 300 soil samples we use. And here you can see that even if the square it's almost 0.8, but you can see that we have very low, very good accuracy. Yeah. So for almost 0.2%. So and this data was generated with using conventional PLS modeling, but we used also for those kind of measurement different kind of algorithm just to improve, in fact, the metrics of those prediction. So thank you.
Well, thank you. Thank you, Christian, and thank you, Jack, for this very wonderful introduction and giving us a flavor of the various and very interesting activities, especially using libs that is currently going on in your company in the field of agriculture. Thank you very much. So with this, the platform is open for questions. I'm sure there'll be quite a few questions. So I think we have already one. Yeah, please go ahead. You are muted. Yeah.
Okay, sorry. Yeah, thanks for a wonderful presentation. And I just have a quick, very specific question. So, because I think Christian showed at like the very first slide he was showing as the technical portion, that you have some sort of a, sort of a soil grader kind of a thing, like you were measuring the particulate size and things like that. Can it also measure soil hydration as well as grain size? Rather grading together, or is it just a sort of that? Am I clear? I mean, what I'm trying to ask.
No, just can you just repeat?
Sorry.
Can you just repeat your question?
Okay. Oh, okay, question. Because I think, maybe I am wrong. I'm not sure. Like because I was trying to get exactly what you were showing. So it seemed like it's you were measuring the soil texture, right?
Yes.
So what I was asking that in doing so, do you only measure the sort of like we say what is the grain size of the soil in general, the average grain size of the soil, or you are also sort of grading there in terms of their the total hydration and maybe even nutrition, like in terms of
Okay,
yeah. I think now I have. Now you have got the question.
Yeah, exactly. So for this one, I just it was we for this question we just limited to actually the texture, but we are able to also measure the nutrient of soil, or the mineral of soil. So in terms of the difference here in terms of for the texture, it's more just to the physical part. In fact, for in fact, it's more like the size of the particle size of the soil, which interact with the laser. This interaction gives us the information about the texture of the soil. Yeah.
So the granularity, you mean to say?
Exactly. So those interaction, we can model those interaction to give us the idea about the texture. So we can go in even to we can classify the subsamples by texture, or we can actually predict the presence of different soil particles. It's about that. So the chemical composition of the soil, this is another application also that we do also.
Okay, thank you.
Yeah. Okay.
I have a quick question.
Right.
You talk about applying this for measuring bulk density. In that case, do you have to use an intact soil, or do you still use a kind of a loose soil sample?
For the bulk density, it's in fact we use the same procedure that we often use. So we have a palette of soil samples, but there is a measurement we have to done before, just to don't lose the information before we go to the palette. So we can measure the mass of the soil, the volume of the raw soil samples. And after that, when we go to the pellet, and with the LIPS technology, we can just combine those information just to retrieve the bulk density.
So the volume will be coming from the field sampling, or the volume of the soil you put into your plastic container in?
The volume, in fact, it's just the volume of the raw. So we have, like, for example, if we sampled different certain amount of soil, in the field, and we go to the lab. And in the lab, we're gonna put those soil samples in different container, and in those container, we have to measure a different kind of in fact parameter just for the raw. So we're gonna measure the mass of the raw samples. In fact, all the things we have to do before to go to the pellet form of the samples. So we don't want to lose so much information about the raw samples. So we measure different kind of the parameter for the raw. And after that, when we go to the pellet, because we have to go to the pellet because for the technology to have accurate measurement, we have to have a pellet. And when you go to the pellet, you're gonna lose some information about the raw samples. So that's why it's two-part measurement, in fact.
Okay, thank you.
Yeah.
Amina, you had raised your hand.
Can I add some? Sorry, can I add something to this answer? Because I'm not sure if Scott Cheng, it was clear. We do a calibration using bulk density measurement the conventional way. So we obtain bulk density results on hundreds of samples, and then we make a calibration and we train our algorithms to find their way to the result. It turns out that bulk density is determined mostly by soil texture and by soil organic matter. So once the algorithms are trained to find it, we don't need to sample for bulk density anymore. We can use the normal sample that we just, like a normal probe, the same sample you use for soil organic carbon, and the algorithms, because they are trained using determinants like organic matter and texture, they predict the bulk density with relative accuracy. I think that's. I think this is where Mr. Chang was going to, but that's really how it's predicted.
Thank you. That's exactly what I was thinking about. But I guess a follow-up question on that is, the bulk density obviously is very much related to the structure of the soil as well. And that has nothing to do with the texture or would probably have something to do with texture and organic matter content as well. But whether you have a soil compaction, which is going to completely change the structure of.
Absolutely.
Yeah.
Yes, absolutely. Yeah. You're absolutely right. When we sample, we've sampled hundreds of fields for the bulk density, and we knew that when we were in a place in the field, let's say they were tractor tracks, and the soil was physically compacted, the libs will not predict the bulk density correctly. It would show as an outlier. And we're able to, because we geoposition our sample, and when we take the sample, our agronomists are able to see that there's there's a particular area that is more compacted, let's say because of tractor tracks. We can easily make the relation between the outlier and our result when we're calibrating. But you're absolutely right. It's it will give us the ball density assuming a normal soil structure, not heavily compacted, not recently fluffed up by tillage. And if we go to outside a certain range that's a heavily compacted soil, it will give us an erroneous result. But still, over hundreds of samples, we were accurate enough that we won the Indigo Carbon Challenge. And one of the reasons is we were able to predict soil density correctly enough that with our percent soil organic carbon, we were able to estimate stock or tons of soil organic carbon per hectare or per acre. But your question is remarkably accurate and correct. Yeah, it's a very good question.
Thank you.
Okay. Thank you. We have time for one more question. So I think Sukhdev has a question. So maybe Sukhdev, if you're here, you can ask the question directly. And please unmute yourself, Sukhdev. Okay, so appears that he's not here, but I'll just read out his question from the chat box. So his question is that he wants to know the comparison of cost of soil testing with traditional methods, including both the setup cost and the running cost of the equipment that you are proposing.
Yeah, that's a good question. I will answer it maybe not quite appropriately, but I will tell you this is when we took the Indigo challenge, this is one of the questions they had, because if it's 3 times more expensive, even if it's faster, we're not going anywhere. So they estimated our impact on sampling. And because we don't need to sample for ball density, we cut the time of sampling by 2/3. So the cost, instead of being roughly $9 per acre, it came down to $3 per acre or something like that. That's the sampling part. Once we bring the sample to the machine, to the lab, you have to dry the sample and you have to mix it, homogenize it, and press it. One operator, once the sample are dry, one operator can basically do a sample per minute, because the time that it presses the sample into the cup, puts it in the carousel, takes less time than actually the carousel being analyzed. So the production cost is very much related to the number of samples you will do, because the machine is expensive. So if you split the cost over just a few samples, it's going to be expensive. But since the machine can do 500 samples with one operator in an 8-hour shift, so it's a sample per minute. If you do 20,000 samples, or if you do 30,000 samples, the cost of analyses, complete soil analyses, including texture and soil organic matter, pH, and so on, comes down to a bit less than $5 US per sample. And of course, the more sample you have, the cheaper it becomes. So that gives you ballpark figures. And that's the numbers that Indigo Ag estimated, I believe.
Okay, so thank you very much. With that, we'll end this talk. I think with this, we'll end this very wonderful talk. I think we are running short of time again. So the questions themselves proved how interesting this talk was. So thank you once again to the entire Logiac team who presented over here for their time out. And we hope to be interacting with you again in the near future. And with that, we'll move to the last talk of this session. And that will be given by Balshed Sidhu. and he will talk about the impact of climate variability on crop yields in India. So to introduce the speaker, he is a doctoral student, actually a PhD candidate at the Institute for Resources, Environment and Sustainability at the University of British Columbia in Canada. And his research focuses on the relationship between climate variability and agriculture using high-resolution spatiotemporal climate and crop yield data from India. So essentially, even though he's doing his PhD over there, it is entirely based on crop yield data and other information from India. And the goal of his research is to propose strategies that can improve crop yields while minimizing adverse environmental impacts. So with that, over to you, Balsher. And so we have 25 minutes. And so what we will do is, just to give you an indication of the time, we will turn off our videos, the organizers, so Prajita, Amina, and myself, and we'll just turn on again 5 minutes before your time ends, so about 20 minutes from now. So give you indication that you have 5 more minutes left. So thank you, and please go ahead.
Hi, hello everyone. Good morning and good evening, depending on what part of the world you are in right now. And thank you for waiting patiently for my talk. My name is Balsher and I'm a PhD candidate. And I'm working on the impact climate has on crop yields in India. And today specifically, I will discuss and compare various statistical techniques that I'm using in my analysis. And before doing that, just a quick note that I'm very happy to be presenting today because I have a relationship with IIT Delhi. I was an undergrad student over there. I completed my BTech in civil engineering from there. So it's nice to talk to people who from my alma mater. Okay, so I'll give you a little background about my research. As we all know, agriculture is highly dependent on climate, both seasonal, like the average seasonal temperature or total rainfall over the season, as well as within seasonal variability in terms of like how many rain days there were or how extreme the temperature was on certain days during the growing season. So in other words, these anomalies that may be overlooked if we only discuss climate in terms of seasonal parameters, these are also very important. And within season variability has been documented to be important for crop yields, and researchers use various statistical techniques for that. These vary from a simple linear regression where crop yields are modeled as a linear function of climate variables to more advanced machine learning methods where huge amounts of data is used to extract relationships, which would otherwise not be possible using linear regression, for example. And in this particular project that I'm working on, our analysis expands along both these axes. So we are comparing seasonal versus subseasonal analysis, as well as linear regression, which we call the simplest model, the simplest method, to other machine learning techniques, which I will talk about later. So some context to my study for people who are not familiar with Indian agriculture. In India, agriculture is heavily dependent on monsoons, which are the summer rainfall. 2/3 of the net cultivated area in India is dependent on monsoons, and it's estimated to produce 40% of the food grains nationally. While predicting crop yield as a function of temperature and precipitation has been studied extensively, research has more or less been limited to seasonal average precipitation and temperature values, and only recently has this within season variability caught attention of researchers. This is important because I'll give an example. For example, with a short drought during the early growing stage, followed by a heavy rain during harvest, if you consider that whole growing season, it will seem as if it's a normal season because there's X amount of rainfall over the season. But the erratic patterns within the season might cause yield losses or even crop failures. So that is a primary motivation why we want to build statistical models that can incorporate this intra-seasonal climate variability to better understand the relationship between climate and agriculture. To begin with, we'll talk about the seasonal model. We have primarily two variables in this, which is the seasonal temperature, the average seasonal temperature, and the total rainfall over the season. These variables we constructed from daily climate data, which is available from Indian Meteorological Department at district scale for all years from 1961 to 2015. And with these two independent variables, we modeled annual crop yields as the dependent variable or the variable of interest. So we have the independent variables on the left side and dependent variable on the right side. This crop yield data was acquired from the International Crop Research Institute for the Semi-Arid Tropics. And this is again at district scale from 1966 to 2011. So my discussion today will only focus on rice for want of time, but we have analyzed 9 other major crops, but we'll only discuss rice today. To give an example of what the output of a seasonal model looks like, if we model crop yields as a function of average temperature and total rainfall over the season, this is what the output of the result model looks like. And the way we interpret these model coefficients from a linear regression is that a 1 degree Celsius rise in temperature would lead to a fall of 0.138 tons per hectare of crop yields. So this estimate over here shows that. And similarly for precipitation, a 1 millimeter rise in total rainfall over the season will lead to a rise of 0.0002 tons per hectare. So the difference in orders of magnitude between these two coefficients rises from the way we define our temperature and precipitation variables. Anyway, so that is how we interpret our linear model. However, as I was discussing earlier, this sub-seasonal climate variability is also important, and even more so due to climate change, which has shown that extreme wet and dry spells during the summer monsoon season are changing. There have been a few studies which have shown that while the mean seasonal rainfall might not have changed, so the total rainfall has not changed, the rainfall pattern has become more erratic, that there are more extreme rain days and then there are more dry spells. So the average rainfall might not change, but the variability of the precipitation distribution has increased. And agriculture is extremely sensitive to such variability. At the same time, we talk about the temperature of impact, and it has been shown to be nonlinear. So a 1 degree rise in temperature from 25 to 26 would have a very different impact compared to a 1 degree rise from, let's say, 35 to 36. And this vulnerability is amplified in India's rain-fed regions, which account for approximately half of the net sown area. So much so that the 5th assessment report of the Intergovernmental Panel on Climate Change, they've ranked Indian agriculture among those most vulnerable to climate change. Now, I would like to take a minute to discuss the concept of degree days. So the way to include the nonlinear impact of temperature on crop yields is to build degree days as a variable. Degree days is essentially, it's a very common method which essentially divides the crop exposure to heat over different temperature ranges. For example, let's say there were 5 days with average temperature of 36 degrees Celsius, and you define your base temperature as 30. So then you see that every day there was a difference of 6 degrees Celsius. So you multiply that with the number of days, 6 multiplied by 5, you had 30 degree days over and above 30 degrees Celsius in that case. So to build onto on a subseasonal model, we added these independent variables, which included degree days and total rain days. For degree days, we divided the crop exposure into 10 degree intervals. So the number of degree days in 0 to 10, in 10 to 20, 20 to 30, and then greater than 30. So that is the independent variable that accounts for temperature variability within the season. And for precipitation, of course, we have rainy days. So these two are standing in for within season variability in our model. Just switching gears for a bit before we return to our statistics. Irrigation plays a very critical role in agriculture, as is common knowledge, since crops receiving irrigation are better suited to withstand dry spells over the growing season. And India exhibits a very large spatial variation in the availability of irrigation facilities. This is shown in this figure, where we have plotted the percent area under rice cultivation that receives irrigation across different states in India over time. So there are some states which have historically had good irrigation facilities, and they were 100% irrigated even in 1980. While there are others which exhibit little improvement over the last many decades, like Maharashtra or Madhya Pradesh. And lastly, there are some which show rapid transition from being primarily rain-fed to being irrigated since the start of the Green Revolution. So because of the strong differences in level of irrigation across the country, I divided my data fed and irrigated, and I'll present my results accordingly. So for this, returning back to our sub-seasonal linear model, I will just give you some sample results for want of time. So showing here on the right side in the plot is the impact of rice getting exposed to more degree days in different bins from 10 to 20, 20, 30, and greater than 30. So what this means is if there are more days in the 10 to 20 degree bracket, so 10 to 20, the crop yields fall. But if there are more days in the 20 to 30 degree bracket, then crop yields improve. And this is for rain-fed areas in India. This impact of climate has been documented in previous research. It has been shown that rice, if provided enough water, prefers warmer climates to a certain extent. But if the temperature goes beyond 30 degrees Celsius, then the crop yields will start to fall. So if we compare these results from our sub-seasonal linear model to our previous seasonal linear model, we saw that a rise in temperature has a negative impact on crop yields. But this analysis shows that the rise in temperature is the impact of a rise in temperature depends on where that rise in temperature takes place. Is it in 10 to 20 degree or 20, 30 or greater than 30? So this is how nonlinearity is accounted for. So we've discussed our seasonal linear model, we've discussed a sub-seasonal linear model, and now we'll move on to machine learning techniques. Machine learning is essentially it's a fancy word for statistics where you don't specify the model. The computer tries to understand the relationship between data without you explicitly telling it if it's going to be linear or quadratic or cubic or whatever. The advantages are that, of course, you don't need to specify a model, so you don't need to start with an a priori hypothesis, and interactions between variables can be accounted for without having to do that manually. And disadvantages are that machine learning models more or less act like a black box. So you input data from one end, you get predictions at the other end, and they are not as interpretable as the traditional statistical techniques like linear regression. So even though their interpretability is bad, there are some machine learning techniques which sort of border between the traditional stats and machine learning, like boosted regression trees, which are sort of interpretable. And that is why we chose BRTs or boosted regression trees for our machine learning analysis. Simply put, a BRT is just a flowchart. So what you do is you input the data into the computer and it tries to bifurcate the data into two parts, and then it continues to do so through successive iterations. So for example, it could look at the data and infer that when temperature is less than T1 and precipitation is also less than T1, then the yield is Y1. Or it could understand if temperature is greater than T1, if it lies between T1 and T2, then the yield is Y3. If it's greater than T2, it's greater than precipitation is greater than P2, then your yield could be Y5. And this is a very simple schematic. A BRT has a lot more trees under it, probably hundreds, if not thousands of these. So because they don't start with any assumption about data distribution, if used correctly, they can handle non-linearity, continue to bifurcate data till they reach a point where further bifurcation does not have any more utility, and then they stop there. So compared to linear regression, BRTs make fewer assumptions about linear, but they also have disadvantages, which I alluded to earlier, and I'll touch again later. I'm presenting I'm just showing one aspect of the results from the BRT again. So my computer is saying my internet connection is unstable. Am I still audible?
We can hear you and see you well. It's okay.
Okay, perfect. So the additional impact that time spent over 30 degrees Celsius, the degree difference that we discussed earlier. So this shows that at the top of the model is irrigated rice yields, and we see that more or less And increasing growing days in the 30-degree bracket does not have much impact on rice yields. But for rainfed agriculture at the bottom over here, shown in red, it is apparent that as the crop spends more time in the 30-degree bin, crop yields start going down. So over here, if we compare this to the linear model, we see that the linear regression model fits a single value. So it says that growing degree days in the 30-degree bracket have a positive or negative impact on crop yields. But a BRT, if needed, it's able to fit a more flexible relationship. And this is more useful in terms of explaining the relationship between yields and climate, which is anything but linear. I'll just present my results in a different way now. It's just a reiteration of the fact that BRTs are able to account for nonlinearity. So the plots show observed yields on the x-axis, so the data that was actually observed, and predicted yields on the y-axis. I just want to clarify here that these predictions are based on out of sample data. So we use 90% of the data to train our model, and then we use 10% of the data to make predictions. So the model has not seen the data that it is being tested on. And then this is done 10 times with different 10 sets. So that is essentially what tenfold cross validation is. So, it's visible over here that BRTs are able to account for nonlinearity better, because as you see over here, as crop yields cross a certain point, the prediction of the linear model actually moves away from the one-to-one line. So for an ideal model, your points would lie on the 1:1 line, but a BRT is able to account for that better. So they are more versatile and robust to the range of crop yields that are observed across the country. And so the plots that I showed in the previous slide, the scatter plots, I've summarized them in this slide. So I'm going to plot RMSEs, or root mean squared error, for both the linear and linear model and BRT, both for sub-seasonal and seasonal analysis. And RMSE, just to quickly explain what it means, it essentially averages the performance of a model over a wide range of data. So you take the difference or the error in prediction for each point, and then you average out that error. So that is essentially what RMSE is. It's the root of the average of the squared errors. And for linear regression, we see that adding sub-seasonal variables has a significant, albeit marginal, improvement on model prediction. So a seasonal model is able to improve our model prediction by around 2% compared to a sub-seasonal model, which can improve it by around 3%. And compared to that, BRTs are improving our prediction by up to 22 to 23% for the seasonal model. And surprisingly, the sub-seasonal model does not perform as well as the seasonal model. So in spite of the massive improvement, BRTs it seems like adding sub-seasonal variables to BRT reduce the model performance. This can be taken as evidence suggesting that BRTs may be overfitting when you throw extra sub-seasonal variables at it. One extension of these results, which I would like to investigate further, is, because this is an ongoing project right now, which I would like to examine further, is can BRTs be used in data sparse situations? For example, I had daily climate data from which I was able to do all this analysis, but if the daily data is not available, let's say there's seasonal data available, can a BRT be used in that situation? Because we see that a seasonal BRT is outperforming even a sub-seasonal linear model. But of course, this is all conjecture that I have only from my analysis looking at crops in India, and I'm not sure if I would like to we need further evidence to claim that if this technique can be transferred to other areas around the world. The significance of my results of my analysis is that we are continuously trying to understand the relationship between climate and crop yields as well as we can. If we are able to predict crop yields depending on within season variability or climate variability, it can inform policy for developing sustainable developing sustainable irrigation facilities in areas which are most susceptible to this kind of climate variability. The government of India has plans to expand irrigation across the country. So this analysis could help us find those low-hanging fruits where irrigation expansion can give us the best bang for our buck. And of course, the results from this analysis can also help us adapt to climate change by not only suggesting better irrigation techniques in areas which are currently rain-fed, but also switching crops where expansion of irrigation is not possible because of lack of water. And that is the goal of this research. And with that, I would like to thank you for listening to my talk so attentively. And I would also like to acknowledge the Vanier Canada Graduate Scholarship for their financial support. This is my email address should you want to get in contact with me, and I can take questions now.
Okay, thank you, Balshed, for this wonderful perspective on climate changes and how they're affecting the crop yields and what can be done. So this forum is now open for questions. So please either raise your hand or you can unmute yourself and ask the question directly. So I think there was a question by Yuxiang Chen. What is the reference temperature for your degree days?
So the reference temperature, so there are two ways to use degree days. Either you can have a reference temperature, which depends on the crop, or you can do what I did, where I had degree day bins. So I calculated the number of degree days in each 10°C bin. For rice, the degree days are usually calculated as anything less than 29°C. So since my model has degree day bins going from 0 to 10, 10 to 20, and 20 to 30, so the addition of all three would be taken as the degree days for rice if we go by the reference temperature. So this is essentially more flexible than having a reference temperature.
Okay, thank you. So if I think Amina has a question since we don't have anything else right now.
Okay, thank you very much, Balshed, for a very nice presentation. It's great to have you here. I was interested in your seasonal model and the data that you intake. What sort of data would you also be interested in having that maybe is not presently available, or in extending this model to different areas?
You mean just the seasonal or the sub-seasonal as well?
Yeah, I guess so. Correct me if I was wrong, but I think you were looking at publicly available data or data that's already collected. And I was curious in your model if there's certain kinds of data that you might have desired that is not currently there. And also it made me think with our first talk that this is a very nice way to inform the kinds of crops that we can engineer that might be more resistant to stress. And so they work together quite nicely. So just curious what other kinds of data you might be interested in looking at and incorporating.
Oh, I see. So since I was looking at data collected in the past, there is only so much I can use. But it would be really wonderful to have maybe soil moisture data, because that is actually how crops react to water. Right? It doesn't matter how much rain falls on the ground if it's not if it doesn't manifest itself in the form of soil moisture, the plants don't really benefit from it. So soil moisture would be great. But we haven't measured soil moisture in the past. We do have some satellite measurements coming in now. The other thing is I would also love to have some non-climate data, like different chemical inputs, because those are a big driver, maybe even more than climate. They are a big driver of crop yields. So controlling for all non-climate data would be really wonderful. And yeah, but maybe crop varieties would be really nice to have, because my model assumes that the crop variety doesn't change over time, or it's the same across the country, and over time it changes uniformly across the country. So it would be really nice to have crop different varieties, like GMO adoption. So sky's the limit in terms of what kind of data we would want.
All right, that's wonderful. Thank you. And I think really goes to the heart of the workshop too. Nice to find ways to pull it all together.
So if anybody else has any questions? Okay, so if there are no other questions, so let's thank all the speakers once more for a very wonderful evening at India and a very wonderful start of the day in Canada. And we hope that this was as enjoying for the participants as we all three of us enjoyed while we put together this conference and these sessions, all the sessions in fact. And with that, we will end today's session. And just before we lose all of you, I would just like to highlight what we have upcoming for tomorrow. So just give me 1 second, just to sort of a recap. So tomorrow we have 2 sessions as we have today. So one in the morning and one in the evening, depending on where you are. So the first session will mostly be dedicated to diagnostic tools for the new age science, where we'll have again actually 4 speakers. So one will be Cody Wilness from a company in Canada, Crop Pro Consulting, and he'll talk about soil and its relevance in the precision agronomy program. Then we are looking forward to a very beautiful talk by Professor Kiefer, where we'll talk about laser wake field driven X-ray sources in Canada. And finally, we'll have a tutorial come live demonstration of agri-photonics or optical techniques or photonics techniques in agriculture, where there'll be 2 faculty members talking about different spectroscopy techniques, one for terahertz spectroscopy and Raman spectroscopy, and we also have fluorescence spectroscopy and other biochemistry techniques relevant to agriculture. So with anticipation for a big, great session ahead tomorrow morning and tomorrow evening, so we end the session today. Good night and have a good day. Thank you very much.